Thank you so much for joining us today. I'm Sarah Taylor, and I'm the Collections Manager for the Herbarium and Paleobotany Collections at the University of Connecticut Biodiversity Research Collections, which we call the BRC. Tonight, the four of us, myself, Katrina Menard, Matt Opel, and Erin Kuprewicz, are going to tell you how the collections were integrated and share a little about each of the collections that we manage. We are here in rural Connecticut, about halfway between New York City and Boston. At the end of the 20th century, the natural history collections at the University of Connecticut were each stored in separate locations, mostly in an aging building with unreliable climate control. The herbarium was full. The Paleobotany collection was nailed shut and covered with plastic because it was stored in a hallway and needed to be protected from theft and leaking pipes. The vertebrates and invertebrate collections were disconnected from the plant collections, and there wasn't a unified vision or much communication between the collections. The greenhouses were overcrowded, hosting both educational displays and controlled research experiments. At the turn of the millennium, the University of Connecticut launched the ambitious UConn 2000 initiative, building state-of-the-art facilities across campus. As part of that program, our Ecology and Evolutionary Biology department chose to unite the preserved collections in a single facility. The university constructed our purpose-built space, and the NSF funded our compactors, equipment, and moving costs. At the same time, the university built a state-of-the-art greenhouse on the roof of our building for botanical research. Less than 10 years later, we united the preserved collections and the living collections, the greenhouses, under the BRC umbrella. This diagram shows the layout of our preserved collection space, with zones for wet and dry storage, preparation and research space, offices, and a collections library. The research greenhouses are on the roof of our building, and the educational greenhouses are just downstairs and across a small courtyard. When you walk into our building, you are greeted by an amazing lobby ringed with comfortable seating areas and lecture halls, and a spacious area to hold community events. This is our main prep room with a shared dedicated space for invertebrate and plant specimen preparation. Our vertebrate preparation area is just down the hall. As you can see, our new area is much brighter. It's safe from pests and has state-of-the-art fire suppression system and its own climate control system. Each section has cabinets designed for its kind of specimens, drawers for vertebrates and fossils, shelves for parasite slide boxes, pigeonholes for herbarium specimens, and slots for invertebrate Cornell boxes. So now I'll tell you more about the herbarium part of our collections. UConn was founded as an agricultural college. Storrs is the name of the village where we are located, and our non-lichenized fungal collection reflects that heritage. The herbarium, after all, was founded in 1898. We have over 200,000 specimens here in the herbarium. We use color-coded folders to reflect specimen geography and currently store bryophyte packets mounted flat on herbarium paper. Our taxonomic strengths are in families well represented here in the Northeast, such as Ericaceae and Rosaceae, as well as species-rich groups like Asteraceae, Poaceae, and Potamogeton. Our type collection is stored in red folders in a dedicated cabinet. Our holotype of Bractiococcus glacialis is our only specimen from Antarctica. While we have a heavy focus on plants of Connecticut and surrounding northeastern North America, we do have specimens from every continent. All specimens have a story to tell, and these are two of my favorites. On the left, we see a specimen of Panicum virgatum collected by Henry David Thoreau in Concord, Massachusetts. And on the right is our only specimen of Bayard's white adder's mouth orchid, Malixus bayardii. It was collected in 1904, and as far as I can tell, is the only specimen of this species collected from Connecticut in any herbarium. It is considered extinct here, and this specimen may be the only evidence we have that it ever grew in Connecticut. Anyone with an internet connection can visit us on the web. From this page, you can learn about our history, search our databases, request a tour, and explore lesson plans and educational resources. For example, you can find a high-resolution image of almost any vascular plant specimen in our collection. If you zoom in on our online specimen images, it gives us about the power of looking through a 20x hand lens. I'm also the collection manager for our Paleobotany specimens. Most of our fossil plant collection was assembled by the eminent paleobotanist Henry Andrews. The fossil leaf on the left is from the Warman clay pit in Tennessee and dates from the Middle Eocene, 47 to 38 million years ago. These fossils are extremely fragile. You can see how friable the clay is. Updating our fossil plant catalog and making the data available online have been a major focus of the last 3 summers. Thanks to the work of Henry Fry, a graduate student in our department, we've documented hundreds of glass slides containing thin sections of fossil types, and we're able to put that information online. And now I'll turn it over to Katrina Menard.
I'm Dr. Katrina Menard, the collection manager for the Invertebrate collection, and I'm going to take you through some highlights of our various diverse groups of spineless creatures. One of the strengths of our collection is the fact that our invertebrate collection is integrated with other collections here at the BRC. There are 3 in particular I'm going to focus on that are very good exemplars of this. The Menard collection, which is also integrated with a Lasso tapeworm database, the Coldwell hummingbird mite collection, and the Rettenmeier army ant guest collection. So the Menard collection is a very large parasite collection. It's especially regional in focus. Dr. Menard used to collect around here in Storrs, Connecticut, of various hosts and the different parasites that were associated with them. So for example, botflies here with various livestock and wild populations as well. So we've got mice and things like that. We also have a large collection of various, for example, beaver ticks and various mites from nests and birds, and a lot of vertebrates that you can't collect from these days very easily. So from a historical perspective, it's very good for recording what we have, but also the fact that we can't necessarily collect anything like these parasites in the future. Another important collection we have is the Coldwell Collection. So this is again a research collection where Dr. Coldwell collected not only data from what kind of hummingbirds that these mites use to travel from host plant to host plant, but also what plants that they are associated with. So various ginger plants and heliconids, and all of these slides that are of the mites are important historical records of, again, what not only these mites use for transport, the hummingbird species, but also what plants they actually reside in once they make that journey. Another important collection we have is the Lasso Parasite collection. This is being currently utilized by Dr. Kyra, who is studying tapeworms that live in the small spiral intestines of various shark groups. So in addition to having a unique and worldwide recognized specimen collection, all of this data is currently being housed on the LR Menard database, which is an online data portal where you can look up not only the different species of tapeworms, but their shark hosts that they are associated with. And this is important for not only studying the diversity of this unique group of invertebrates, but also, again, sharing how this data is linked and important for crossing different groups. Our last integrative collection that we're going to highlight is the Army Ant Guest Collection. This was donated by Dr. Rettenmeier, and it was integrated through a National Science Foundation grant to digitize not only the specimen data we have of the army ants, but their guests, which accompany these different species, as well as other data sets which are not usually linked to specimens. For example, Kodachrome slides, so different images, as well as field cards and other associated data from the scientists as they were observing these unique insects. All of this data, thanks to the NSF grant, is also available online on our database, the AAGC database, where the public can peruse the different ways, again, this diversity is linked, and also, again, these unique images that were taken as part of this research study. In addition to the integrative data sets, we also have various taxonomic strengths. So various groups of insects historically that are linked to professors that worked on these groups or associated scientists who deposited their material here. For example, we have one of the best dragonfly collections in Connecticut. This is part of work done by Dr. Wagner, who created a species list to investigate what endangered species we have or what species of dragonflies are threatened in our state, as well as across New England. Historically, we've also had a strong research program in Heteroptera, so Dr. Slater and then Dr. O'Donnell and then myself, Dr. Menard, we all have done research in Heteroptera, and therefore that expertise has been represented in our specimen holdings. Lepidoptera, so butterflies and moths. So again, this is thanks to Dr. Wagner and a lot of his colleagues that work on moths and butterflies. We have a strong bee collection. This is part of the stage collection as well that was deposited here. So we have a very strong regional bee collection for North America. And again, we also have a strong research focus on tiger beetles, not only because, again, they're fairly charismatic, but they're also endangered and threatened across many areas in the United States. And specifically, there's the federally listed Puritan tiger beetle, which is shown here in the illustration, that is only known from a few populations along the Connecticut River here in New England. We also have a very strong regional strength for New England taxa. As I mentioned before, a lot of what we know about dragonflies and damselflies of New England and Connecticut is thanks to the work that Dr. Wagner has done, as well as, in various field expeditions across the New England area to deposit specimens here. So if anybody wants to know about the New England fauna of these groups, they're, of course, very welcome to come and look at our material. As I mentioned before, we also have the stage collection of North American bees. This diversity is also one of the reasons we're applying for an NSF grant to digitize our bee collection, because it is so well representative of different groups across North America. And again, we also, because we have such a diverse sampling of New England taxa, it's possible to kind of record what species are no longer present or what species may be threatened or endangered as habitats change thanks to global warming or human development. We can come up with indicators as to which species are no longer in the same habitats anymore, if they're even around anymore, maybe possibly extirpated.
Good afternoon. This is Matt Opel, the collection scientist at the University of Connecticut Plant Biodiversity Conservatory and Research Corps, which includes the living plants used for teaching, research, and public outreach at the UConn Biodiversity Research Collections. The Plant Biodiversity Conservatory includes 2 separate greenhouse facilities dedicated to teaching and to research. The permanent teaching collections are some of the most diverse at any U.S. university and include something on the order of 1% of all described plant species. The research collections are used in projects that have brought in $10 million in external funding to the university in the past 11 years or so. In the greenhouses, we utilize integrated pest management and other modern greenhouse control practices. And the greenhouses have a very strong web presence with detailed information on all of the plant accessions, as well as legally required pesticide and other records. The collections are well utilized in teaching. with nearly half of undergrads accessing the greenhouses as part of their coursework in an official capacity at some point during their time at UConn. The biodiversity collections are open to the public as well for tour groups, and we also give presentations for various public organizations. We provide plant material to external institutions in the US as well as internationally as permitted by regulations on a pretty regular basis for research or conservation related projects. In the second half of my segment, I'm going to do a short introduction to 3 of the outstanding collections at the UConn greenhouses. Our holdings in the order Zingiberales were recently augmented by the acquisition of a duplicate of the John Kress collection, which was at the Smithsonian Institution. This collection now includes primarily material with excellent wild collection provenance, including some new and undescribed species. These are on display in the teaching greenhouses now, and there are several researchers who have expressed an interest in using them in their research work in the future. The university is also home to a remarkable collection of species Pelargonium, and these are the wild relatives of the windowsill geranium. This is a plant group that's primarily from South Africa and includes familiar herbaceous species like the windowsill geranium and scented geraniums, as well as more unusual forms like winter-growing geophytes and highly xerophytic succulent plant forms. The university's collection in the dwarf succulent genus Conophytum, which are sometimes called living stones, is one of the best in the world. The collection is very close to complete taxonomically and is extremely well documented with wild collected material. These are plants that originated in arid winter rainfall habitats in southern Africa, Namibia, and South Africa. The Conophytum collection includes a number of species that are of conservation importance. So many populations of Conophytum are in steep decline in their native habitats because of recent record-breaking droughts and also a terrible increase in poaching for the succulent plant trade. The plants pictured here are either extinct in the wild, known to be extinct, or critically endangered and probably rapidly heading towards extinction. So the UConn Living Conophytum collection is of significant importance to conservation efforts. Thank you for listening to this short introduction to the Plant Biodiversity Conservatory at the University of Connecticut.
Hi everyone, I'm going to lead you today on a virtual tour of the vertebrate collections that we hold in the Biodiversity Research Collections, or the BRC, at the University of Connecticut. I'm Dr. Erin Kuprewicz, and I'm the Vertebrate Collections Manager, and I'll be leading your tour today. So the scope of all of our vertebrate collections is global, but we do have some standout collections within each vertebrate collection that kind of highlight certain geographic parts of the world. When I'm talking about vertebrate collections, we're considering 4 sub-collections within our division, which are mammals, birds, fishes, and herpetofauna, reptiles and amphibians. So regarding birds, we have amazing representation from the New England states, from the northeastern USA and Connecticut, of course, where we're housed, but we also have just good representation throughout the world in birds. Regarding mammals, we have, again, excellent representation from the US, but we also have an amazing collection of Paraguayan mammals, in fact, the largest collection in the world. For fish, we have great Connecticut species housed in our wet collection, and for herps, we have a really strong collection of neotropical herpetofauna, mostly frogs and amphibians. I'll first talk about our mammal collections. You can see here an open drawer of Zapus hudsonius, a jumping mouse native to the New England area of the United States, And we have round skins here, one flat skin, and associated skeletal material. In our mammal collections, we have over 12,000 specimens. They're mostly kept as round skins, again, which you can see there, with associated skeletons. We also have skeletal material kept separate from skins, and we have fluid preserved mammals as well, mostly bats and small rodents in the fluid collection. The UConn BRC has we're the largest repository of Connecticut mammal specimens in the entire world, which is really special. And all of our data in the mammal collection is available for you to look at right now through the data aggregator site VertNet. So check that out if you want to see some more information and potentially use any of our specimens in your own research. I mentioned previously that we have really great representation of the mammals from Paraguay, especially from the Chaco region in Argentina, Paraguay, that part of South America. This is a standout specimen from our Paraguayan collection, this paratype of Cabesus chacoensis, the Chacoan naked-tailed armadillo. We have all parts of this paratype. You can see skeletal material kept here. We also have material kept in ethanol. And we have the shell of the specimen in our dry collection. So it's really special to have a paratype mammal, especially from our standout Paraguayan mammal collection. Moving on now to our bird collections. Again, we're global in scope here, but we have really strong representation, of course, of New England birds. In our entire bird collection, we have over 10,000 specimens, again, mostly kept as round skins. We do have some flat skins. We also have an egg collection, a nest collection, and a feather collection from which people can take feathers and sample them destructively. UConn is actually the largest repository of Connecticut-based bird specimens in the entire world, which we think is pretty special. Our bird collections are probably the most active of all the 4 vertebrate collections in terms of research, student use, outreach, that kind of thing currently. We have several projects running right now. One of the highlights is working with the Avian Records Committee of Connecticut, or the ARCC, to compile photos of rare specimens along with historic field notes from treat, from sage, from these naturalists from the 19th century. And in compiling data for this project, we were able to actually find records of 12 individuals that were first collected for the species in Connecticut. You can see some of the images from our dry collection here, some of our cabinetry. Here we've put out a lot of our specimens for the records committee to use and photograph. And this is just so you can get a little bit of a sense of our space on the virtual tour and how we can use it when different groups come in to use the bird collection. Moving on now to the fish collection. Again, we have over 10,000 specimens, mostly preserved in ethanol in our wet collection, but we also have several skeletons for comparative anatomy use. We have over 700 species represented, which were used in creating the Freshwater Fishes of Connecticut and Saltwater Fishes of Connecticut books. And finally, our last collection, the Herpetofauna collection. We have about 5,000 specimens, all preserved in ethanol in the wet collection. Most are from Connecticut and the Northeast USA. Are you sensing a pattern here? But we do have significant holdings of frogs from the neotropics, as well as lizards from Puerto Rico and South Africa. Now, we don't really go out on collecting expeditions currently to add to our collection. We collect and grow our vertebrate collections via salvage. So taking animals that have died of natural causes out in the world and having our colleagues and friends bring them in if they're in good condition. They can fill out a salvage slip, which you can see here for a couple of specimens that were brought in to me. And I've hidden the information to keep identities safe and private. But you can bring these in to me. I have permits to collect under our salvage with the government, and I will eventually turn birds and mammals that we mostly get from the wild into round skins to incorporate into our collection. And we actually, before COVID, the COVID era, we had a really thriving and dynamic skinning club where undergraduate students from the Ecology Evolutionary Biology department, faculty, staff, graduate students, all would come in on Friday nights to skin birds. And this really helped me with the backlog that we have in our freezer for our collections. And it really provided hands-on training that isn't usually available to students in any of our programs in museum sciences. So they could learn how to actually incorporate and skin birds and incorporate them into our collections. It was really special. We hope to resume once things get a little bit more normal around here. So that's it for our virtual tour. It was short and sweet, but if you have any questions or you would like to contact me for a visit, for a tour, or for research using our vertebrate collections, there's my email. You can also find me through the BRC website in the University of Connecticut. And it was really great taking you along on this tour. Have a great one. Bye.